This video will be a brief introduction to mapping, which will be continued tomorrow in more detail, but for now we're just going to go over some general principles of mapping, some theoretical considerations to give you a ground to work from. And so, what is mapping? Mapping is basically a subtype of sequence alignment, so a specialized form of it. So some of you may have previously done sequence alignment, for example, with ribosomal genes. So perhaps you amplified a ribosomal sequence, sequenced it using Sanders sequencing, and then you wanted to align it or a number of others to sort of a reference sequence, find differences between them, perhaps construct a phylogenetic tree. So this kind of sequence alignment, multiple sequence alignment, is sort of the classic form where you have some number of sequences, two or more, that are all basically the same length approximately, and we want to line them up, right? Well, mapping is when we a case where we have many small sequences, in our case, sequencing reads, and then we have one or more large sequences. So this can be chromosomes in a genome, parts of the transcriptome, a number of things. The basic structure is that we have a lot of small reads and a lot of small sequences and one very large sequence, and we want to find the best alignment of all of those small sequences to those large sequences. And this really requires a different approach than classic sequence alignment. The algorithms that you use for classic sequence alignment, like BLAST, you're really not going to use for this, and we're going to have completely new software that we're putting to work. And so there are a few kind of theoretical points I want to touch on here. One is end-to-end versus local alignment. So this is a choice that you'll generally have to make when you run read mapping software. And in end-to-end mode, all bases in a read are aligned. So the alignment has to involve every single base from one end to the other in the read, in the short sequence, and we can't get rid of any of them. In local alignment, we can actually trim or soft clip bases at the end of a read. So what that means is that if we find through some algorithm that the alignment is improved by taking out some number of bases at either end of a read, we can sort of take those out of consideration in the alignment and align with the remaining center portion of the read. And so you're going to have to choose these, and of course there are various scores, settings, parameters associated with each of these, especially with local, that you can set. They'll be covered in more detail tomorrow. I also want to point out the distinction between color space and base space. Now, chances are you won't encounter this because most sequencing platforms, including Illumina, which you'll probably be using, use base space, but there is one technology, solid sequencing, that uses color space, so I just want to make sure that you're familiar with it. So base space is kind of what you would expect. A read is given to you as a sequence of A, G, C, and T, just as they were read off from the sequencing. But ABI Solid works a little bit differently. Instead of reading off bases, it reads off transitions of bases. So, for example, we read off the transition from A to C, or A to G, or A to T. And then in the next position, we have the transition from what was the second base becomes the first base, and the third base is now the second base, and we have a transition from whatever that base was to this new third base. And basically, this represents what the platform actually sees, and it is possible to do a lot of your analysis, including mapping, in color space without converting to base space. And generally, converting to base space can introduce error, so you want to remain in base color space as long as you can if you're working with this kind of read. And certain mapping software can use color space and other programs cannot, and that's something that will be covered in more detail later on. And I just really want you to know that this exists for future reference. So we need a way to quantify how good an alignment is and to score it so that we can basically rank different possibilities, compare, go after this kind of thing, and say, in the end, how good is our alignment. And so this is actually quite similar to Phred scores, but we're calculating per read. So we take the probability the read has been aligned incorrectly, which is P, And that's really calculated in a way that's unique to each program. But basically, it's going to depend on the read base quality, read length, how unique is that mapping that we've found, how unique is that alignment within the genome, and some other factors. So we calculate some probability p that the read has been aligned incorrectly, take log 10 of p multiplied by negative 10, and we get out q, which is our alignment score for that read. And a higher score indicates a better and more unique alignment. So basically, higher quality reads, longer reads are generally going to be more unique within the genome, and therefore will have a higher score. As you might expect, if you're mapping to a repetitive region, something like that, you're going to have generally lower quality alignment. And I just want to point out an issue here, which is that we can really blow up the number of possible alignments very, very, very quickly. So if we just have 10 million 100 base pair reads, and we're mapping end-to-end to the 5 million base pair coli genome, we very quickly blow up. So you can think that each, and this really isn't a very high number of reads, right? But still, each read can be mapped to the first base pair of the genome and 99 base pairs downstream of that, or to the second base pair and 99 downstream of that. So for, as you can see, for each of those we have 5 million minus 100 possible alignments, and then we have 10 million reads. So immediately we have 50 trillion possible alignments. And this is already getting really out of hand and past the point where we can just say, okay, we're going to manually check every possible alignment. You can imagine if you allow soft clipping, if you allow gaps or any of these kinds of things, then that just blows up even more quickly. You add more reads, you have a larger genome, it all just blows up exponentially beyond here. So it's impossible for us to actually check every single possible alignment. And so what mapping software does is it takes what's called a dynamic programming approach. And basically what you do is you take what's called a seed. So this is a substring of the read, some portion of the read that we pull out. And then we map that seed to begin with. And having mapped the seed, we try to extend outwards from a good mapping of that substring. And so we try to add on more bases on either side of the seed and extend that alignment. And that's the basic approach. And as you can imagine, there are a lot of parameters that we can set in this process. And you'll need to consider that in many cases. What are the ideal parameter settings? And there's also a lot of considerations if you're using paired end reads. So first of all, you need to tell the relative orientation of mate pairs in your library. So you have read 1, read 2. Are they going in the same direction or in opposite directions? And are they on the same strand or opposite strands? So you'll need to be able to tell the aligner that. And you may need to give the expected separation of the read. So what's the distance between each read within the DNA insert? And of course on the genome once mapped. Though often these days software is able to calculate that itself, though you may need to give it anyway. And so with this information, the aligner is able to take into account where we think the read should be and help put that into the calculation of how good an alignment is, and to help search the space of possible alignments. And we can also use this to map over repetitive regions sometimes to say, okay, so we are mapping with one mate pair outside of the repetitive region successfully. Well, that tells us that this other one must be in this general area of the repetitive region, right? So that can be quite useful. But one consideration is if we can map only one of the two pairs, should we use that unpaired alignment or just from that entirely? So that's often something that you'll have to define in software, that choice. So just something to keep in mind. And now I just want to do a quick overview of file types. And so we have kind of 4 basic types of files that you can deal with. And one is just plain old sequences, and here we have FASTA. Then we have sequences with associated quality scores, and that's going to be FASTQ format. Then we have alignments of sequence of reads to a genome, and that's going to be in the SAM or BAM format generally, though there are quite a few others. And then there's annotations, so basically the genes that we think are on a genome. And that's generally going to be in the bed format, though there are many other formats available for annotations. And so in the FASTA, we have a plain old text file, and we have this caret indicates the start of a sequence, and specifically the start of the name of some sequence. And then we have another line, and on the next line we have whatever that sequence is, and then we can have another line and another sequence, and so on and so forth. And a key point here is that these sequences can be DNA, RNA, or protein. So there's some flexibility there. In FASTQ, things get a little bit more complicated. So we're including quality scores, right? So the start is going to be, we're going to have an ampersand symbol, @ symbol. Then we're going to have some sequence ID, some ID of, generally in our case, of the particular read that we're working with. We have the bases associated with the read itself. This can be in color space in the case of solid sequencing. Then we have a new line, we have a plus symbol, another line, and a series of symbols that are matched up exactly to the bases here. And so each of these corresponds to a specific quality score, a specific thread quality score for that base. So this is the quality score for that base, and this is the quality score for that base, and so on. And so once we've aligned, what we end up with are SAM or BAM files. So this here is an excerpt of a SAM file. And really the details of what this looks like are not that important, but what you need to know is it tells you where is each read aligned, what is the sequence of the read sequence is aligned to, how good is this alignment using the alignment quality score we went over earlier, what strand is it on, things like this. And then we can basically, a SAM file is a text file. It's quite large, generally speaking, and a BAM file is just a binary, so not a human readable file format, but a binary compression of the SAM format that takes up less file space and can be more efficient to process. So it's fairly common to have to basically convert SAM to BAM at some point in analysis pipeline. And finally, we have a bed alignment file. So this is going to be a tab-separated text file with a number of columns that define genes in a genome. And so the first line in the file is going to just define the track name. So whatever our alignment track is, we give it a name, description, whatever else. This isn't actually necessary, it's optional. And then we have 3 mandatory columns. So the first column is just what chromosome are we mapping to. The second column is the start site on the chromosome of this gene, and the third column is the stop site. So that we absolutely must have. And the remaining 9 columns are optional. So we have the name, we have a score. This is kind of arbitrary, it depends on what your application is. Then we have strand, we have the start codon position and stop codon position. So if we're dealing with a situation where, start and stop codon can differ from the start and stop of the gene, this just defines that. And then we have the number of exons. We have the display color, which basically we can color different genes or different annotations to be different colors in a visual display of this annotation. And that can be useful for looking at categories, basically. Then we have the number of exons, comma-separated list of exon sizes, and comma-separated list of exon start sites. So we're able to give some idea of the structure of the gene in a bed format as well. And then each line is just a new annotation, a new gene, using the same exact format. And so this can obviously go on for quite a while since, in the case of any genome, pretty much, you can have thousands of genes, of course. So these get quite large. And this is a format you'll likely be working with quite a bit if you're doing this kind of work.